Okay, my thesis project aims to develop a novel therapy for treating malignant melanoma cells by using H-short protein inhibitors. And more than 50% of melanoma cancers are caused by ROCK mutations. And ROCK inhibitor was approved by FDA to be used clinically and has achieved several success. However, this success is limited by the development of drug resistance. And several research shows that the use of inhibitors of both ROCK and PI3K-AKT cell proliferation pathway can delay the emergence of drug resistance. However, this was not successful due to the development of drug resistance to both the ROCK inhibitor and AKT inhibitor. In order to, the figure shows the tumor of melanoma cancer patient, and after treatment with the ROCK inhibitor, you can see that the majority of tumors disappeared. However, several months later, you can see that the melanoma cancer cells began to be resistant to the ROCK inhibitors, resulting in the formation of new melanoma cells. And in order to prevent the drug resistance, we choose the H-short protein inhibitors as a group of new drugs to treat melanoma cells. And H-short proteins are a group of important chamfer proteins which assist proteins to fall into functional structure. And H-short proteins family have been found to be overexpressed in several cancers, suggesting the critical role of H-short proteins in tumor initiation and tumor formation. Based on these scientific findings, we hypothesize that the H-short protein inhibitors can be used as a novel anti-melanoma drug to effectively kill melanoma cells. And there are two objectives in my project. Firstly, we study whether the H-short protein inhibitors alone can inhibit the growth of melanoma cells. And secondly, we want to examine if the combination of the ROCK inhibitor and AKT inhibitor and a H-short protein inhibitor can achieve a better therapeutic effect against melanomas. And the ultimate proposal for this project is to develop a H-short protein-based therapy for treating melanoma cells. That's all. Thank you.
Good morning. Our research aims to characterize the fluorescent DNA polymerase for PCR. PCR is a technique used to amplify DNA molecules into large amount, which can be analyzed and manipulated. From the first figure, we can calculate that after 30 cycles, there will be about 1 billion DNA molecules only from a single copy. The basis of PCR is Taq polymerase, which is responsible for copying DNA in PCR. This enzyme is the most stable and is still active under high temperatures that are required for PCR. Some fluorescent protein will also have the ability to refold after heating and cooling cycles. This property enables us to combine the Taq polymerase and the fluorescent protein together to generate a fluorescent tag, which is very useful in studying PCR process. That figure is a structure of green fluorescent protein. Within the beta barrier-like structure is a fluorophore. The fluorophore absorbs UV light and emits visible light. The DNA sequences of Taq and fluorescent protein were connected to generate a fusion plasmid DNA. This plasmid DNA was then transformed into E. coli, and we use the E. coli to synthesize our target protein, fluorescent tag. The successful expression and isolation of the fluorescent tag was indicated by a single band with specific molecular size in SDS page. The fluorescent tag was then concentrated. We got only 100 microliter of this enzyme from 1 liter bacterial culture. We then run PCR to check its catalytic activity, which was shown as which was indicated by quantity of DNA products, shown as a bright band in DNA agarose gel. Its refolding ability was also determined. We measured its fluorescence under different temperatures. When the temperature increased from 25 to 30 to 40 and finally to 80, the fluorescence decreased. But when the temperature returned to 25, the fluorescence also returned. We then can use the Fred experiment to study the process of PCR. We will also prepare a pair of fluorescent primers. When the primer binds to the DNA template and are close enough to the fluorescent tag we isolated, the energy can be transferred from the donor to the acceptor. By detecting the signal light emitted by the acceptor, we can know that when they are associated to start copying DNA molecules, and when they are dissociated and stop working. In this way, you can study the process of PCR. Thank you.
My thesis was on the fauna and depositional environment of a late Pennsylvanian fossil vertebrate locality in southeastern Kansas. The locality itself is in eastern Greenwood County, which is about an hour south of Emporia, as you can see in the map at the top corner. The aerial photo just below that is of the site itself. Those two lines indicate the boundaries of the area where most of the fossils were found. The unit of interest was the Snyderville Shale, which is Pennsylvanian in age, about 300 million years old. Interesting to note, this is one of only 3 fossil vertebrate localities in the state of Kansas of this age. The other 2 are marked on that map. The area itself is a hillside. It's actually a railroad cut. The top of that railroad cut is capped by these sandstone blocks. These red lines denote a feature known as crossbeds, indicating that the sands were initially deposited in a fluvial channel, a river channel at some time. Specific identified taxa found were orthocanthus sharks. That's the top left there. The lungfish, Saginotis. Basal amphibians of the family Tendispondyle. Reptiliomorphs, which are transitional animals between reptiles and amphibians and reptiles from the family of Emballomeri and Diadectomorpha here. And synapsids, which are reptile-like animals on the transition to mammals of the families Ophiagodontidae and Sphenagodontidae. The presence of primarily the amphibians and the synapsids, along with the fluvial sands, indicate that this was a fluvial floodplain at some point. The fossils were probably disarticulated in the stream channel itself, and then deposited in that area, probably in a small depression, probably by a flood event.
Our lab is working on melanoma, which is the most aggressive type of skin cancer. It causes most of deaths in skin cancer. About 76,000 new melanoma will be diagnosed in 2016 in the United States. 1 out of 50 persons will develop melanoma in their lifetime. The 5-year survival rate of melanoma is less than 20%. So looking for an effective therapy for melanoma is very urgent. The studies show that about 50% patients with melanoma have acquired BRAF mutation in a gene. Fortunately, current FDA-approved inhibitors have shown remarkable anti-melanoma activities in these patients. Okay, so in these patients, however, most patients developed drug resistance during the treatment, leading to tumor regression. My project is to overcome drug resistance and to prolong the survival time of patients. To achieve this goal, we are going to looking for the molecules that can be added to the BRAF inhibitor to yield a cocktail therapy. Autophagy is the process that cells themselves to deal with the stress, such as lack of nutrient or exposure to toxic substance. An analogy is that you will eat your arms, try to survive when you are extremely hungry. So autophagy helps cancer cells survive and grow. So if we can inhibit autophagy, melanoma cells will not be able to use autophagy mechanism to survive. Therefore, my project is to screen autophagy inhibitors which are most promising candidate of a cocktail therapy for treating melanoma. We would like to first examine whether autophagy inhibitors can suppress the growth, migration, and invasiveness of melanoma cells. If they do, what are the possible working mechanisms? We also like to examine whether autophagy inhibitors can prevent the drug resistance in melanoma cells. This proposed work will lead to the development of a cocktail therapy that is more effective than current single drug treatment and can prevent drug resistance in malignant melanoma. Thank you.
Hello everyone, I'm Huiyan from Dr. Yan's lab. My thesis is about DNA repair system in bacteria E. coli. DNA mismatch repair is a critical supervision system for recognizing and repairing wrong insertion and deletion of base. Major component of the system consists of 3 proteins: mutation, mutal, and mutase. Mistakes only occur on newly synthesized strain. So, DAM is a gene that can add methyl group to the parental strain and tell the system which strain to repair. Since mutase recognizes the mismatched site, which is the first step of repair process, we hypothesize that defect in mutase result in more mutations than defect in mutation. Using E. coli that is not able to synthesize tyrosine, the mutation frequency can be calculated by dividing the number of mutations by the total population of the cells. Our data show that defect in mutase caused more mutations than defect in mutation. Interestingly, double defect in DAM and mutase synergistically produce a much higher mutation frequency than the summation of mutation frequency caused by gene defect in mutase and DAM individually. Therefore, in conclusion, mutase defect leads to higher mutation frequency than defect in mutation. And DAM may work with mutH and mutal to partially recognize the mismatched site. Or maybe DAM is involved in other repair mechanisms that do not depend on the mismatch repair system. Future study may focus on how DAM cooperate with the mismatch. The mutSL is complex in the mismatch repair system. What is the significance to study these genes? Most cancers result from mutation, so it is critical to understand how DNA repair works in the body. For example, impaired function of DNA repair genes may lead to a variety of cancer, such as colon cancer, malignant glioma, and skin cancer. Therefore, studying these genes may help us to design a novel cancer therapy and hopefully save some lives. Thank you.
Today, I'd like to share with you my thesis research about P53 and melanoma. To gain a fashionable tan skin or get more vitamin D, you may like to take sun bath. But do you know the UV in the sunlight can cause DNA damages in our cells? But don't worry, there is an important bodyguard called P53 in our cells, which can help us to repair those DNA damages. The P53 can control the expression of a bunch of genes in our cells. If DNA is damaged by the UV light, the P53 will command the genes to repair those DNA damages. Hey, you guys, start work! If the damage is really severe, which cannot be completely repaired, the P53 will activate the expression of the gene that triggers cells to commit suicide. Thus, the P53 can prevent mutations in cells that potentially transform cells into cancer cells. However, on the other hand, the P53 has its dark side. If P53 doesn't work, the P53 network will be completely messed up. The P53 network will be totally messed up. The damage will not be repaired. The mutations will occur. The cells will develop into tumors. Therefore, like a Taiji, P53 can act as yang, which prevent cancers, and also act as yin, which promote cancers. Melanoma is a type of skin cancer. Many melanoma cells have mutations in P53 and a gene called BRAF. In the context of mutated BRAF, we want to compare the activation sequence and expression levels of the genes in control of P53 in response to UV radiation between melanoma cells with non-functional and functional P53. We will also compare the expression, the activation of P53 network in normal skin cells with melanoma cells. By knowing the differences and understanding the abnormal behavior of network of P53, we want to identify the genes that we can target to restore the regulation and functions of P53 network. This work may bring insights for us to develop novel drugs for treating melanoma. Thank you.
Maternal stress is a concern for many expecting mothers. This is reflected in the market, a booming market that includes books, articles, and even specialized yoga programs, all aimed at reducing maternal stress. So our goal in this study was to see how maternal stress affects, or psychological maternal stress affects the incidence of cyclophosphamide keratogenesis. And so we first became interested in this after we came in contact with it at a meeting. We got interested in maternal stress, and I had been studying previously how antioxidants can curb the cyclophosphamide keratogenesis or cyclophosphamide-caused birth defects. And so we then wondered, well, what will maternal stress do to our cyclophosphamide birth defects? So after literature search, we found relatively few articles with cyclophosphamide and birth defects, and none with our drug. So we decided to go ahead. How do you stress the mice? That's a huge question. So we ended up using restraint stress, in which the mice are confined for a period of time. That makes them uncomfortable, it doesn't harm them, and this causes psychological stress. Okay. So as you can see, what we found is not what we expected, as suggested by the title. And so we had hypothesized, as you probably presume, more stress, more birth defects. And so before we can discuss the result, we need to talk a little bit about the experimental design. So there's 4 treatment groups: control, stress and cyclophosphamide, and cyclophosphamide only. The cyclophosphamide is the drug. You might be wondering, why do I need a drug? This is supposed to be about stress. The drug is so that we can observe the birth defects. If we were to wait around for an event that happens 1 in every 1,000, 1 in every 100,000, we'd never be able to observe it in the lab. So we have to artificially raise the flow of birth defects. We then compare the rate of the drug and the stress, and then the drug alone to see the result of stress. So you can see here that the pink bars are the drug only, and the blue bars are the drug plus stress. Something's wrong. These are flipped. They're not flipped in actuality. This is what we found, but it's not what we expected. Remember, our hypothesis was that the stress would make the effects of the drug worse. We don't have a mechanism elucidated. We don't know what exactly causes this, so we need more study. Currently, I'm doing skeletal data, so I'm collecting data on to try to find rib malformations and stuff like that. So we can see that those are not evident in the fetuses at this level, this gross observation level, in which we're observing head, tail defects, and eye defects, stuff like that. So they're actually skeletalized, and we look at them under the microscope like this. Okay. Overall, this study begs a lot of questions. As always, we have more questions and answers.
Good morning, ladies and gentlemen. My name is Matt Burrows. I wrote a thesis entitled Bocord, America's Blind Date with Voodoo and H.P. Lovecraft's Eldritch Zombie Literature. Within these pages, I discovered or detailed a comparison between 2 different interpretations of the zombie, the Haitian zombie and H.P. Lovecraft's zombie of the early 20th century. These types of zombies are not the flesh-eating zombies that you typically think of. Rather, in the Haitian case, there is a zombie that is created by Haitian Bocord, which is a voodoo sorcerer conjuring in black magic. In H.P. Lovecraft's zombie, detailed within the cases of The Outsider, which was published in 1924, Herbert West Reanimator in 1921, and The Thing on the Doorstep in 1933. All of these zombies are created by either science or by spiritual awakening. What Lovecraft attempted to do with these types of stories was to conflate his own fears about racial integration, about the own fear about oppression, or about the changing of cultural traits within America. In the Haitian version, it is the Vodou sorcerer who's conjuring in black magic, but it is also equally about oppression because it is a reflection of Orientalism in that there is a manipulation of both knowledge and a mastery of each person, not detailing socioeconomic differences. In the conclusion of this research, I found that in both cases there are significant fears about social stratification in that when we created a zombie in terms of the fiction archetype of it and the ones that have continued onward, and is a reflection of the fears of what we do not understand. It is a reflection of the fear of both racial and social changes that are occurring within our culture. Why is this research important? Because it reflects a sort of better understanding not only of our own culture, but of the differences that separate us from everyone else. Thank you.
Good morning, my name is Rosalie Prater, and today I'm going to talk to you about my thesis called Salted Earth, an original fiction manuscript, and analysis of the monstrous female archetype. Western culture, American culture specifically, practices an anthropocentric logic of domination, which implies that not only are humans more important than nature, such as plants and animals, but we should be dominant over them because of that. For something to have value in such a culture, for nature to have value in such a culture, it needs to be fruitful or productive for us. It needs to provide us with something. For example, land needs to provide us with crops or food or shelter, etc. If it's not useful, then it can be sacrificed for something else. You can build a parking structure there, what have you, it doesn't matter. The problem is that we start to talk about gender the same way we talk about nature. Men are seen as more logical, more cultured. Women are seen as more nurturing and more emotional. We are essentially seen as closer to nature. Because of that logic of domination, however, we see that men are on the level of humanity and women are on the level of nature, which implies that men should be dominant over women. In addition to that dominant structure, that implies that if women are not useful, just like nature, then they can be sacrificed for something else. They are useless. In my thesis, I started to look at literature to find ways that that is reinforced. And I found that it's reinforced through certain types of tropes, or how we portray childless women in particular. Because the main reason that women produce for society is through our wombs, by reproducing. Even the language that we use to talk about reproduction is linked to the way we talk about land and nature. Women are either fruitful or fecund, or they're barren or useless or arid, etc. The problem with this is that it marginalizes a large group of people, and that the stories we tell our children, beginning with folk and fairy tales, further enforce this idea. The women in these stories that are childless, for example, the wicked witch who lives in the woods, or the evil stepmother who, comes into someone else's family, reinforces to children that this logic of domination is both okay and normal. It naturalizes it, which pushes an entire, already fairly silent group of people further into the margins.
Uranium-235 fission energy and daughter pair products. The purpose of our research was to examine the mechanics and the potential growth in the fission process. Fission is a process in which a large nucleus such as uranium-235 splits into two pieces, and in the process releases a large amount of energy. This releases about 10 million times as much energy as the combustion of a gasoline molecule. Now what we found was that uranium-235 will split into, there's a potential of 383 different isotopes, which you can split into, each of which has a unique energy associated with it. What this means is that depending on how the nucleus breaks up, there will be a unique amount of energy released. For applications of this, it means that for nuclear energy, if you can select the unique way that the nucleus breaks up, you can select the amount of energy that is produced, and if you can choose the particular products that produce the most energy, you can reduce the amount of fuel that is required. You can also use nuclear medicine. When the nucleus breaks up, some of the isotopes that are produced can be used in nuclear medicine for diagnostics and also the treatment of cancer. If you can choose what these particular products are that are produced, we can also choose to produce more of these nuclear medicines that can be then refined from the fuel to turn the spent fuel instead of a waste into now an asset that can be sold as nuclear medicine. Thank you.
Hi everyone, thank you for being here today. I'm going to talk about Chrysemys picta, or the painted turtle up on the upper left, and how it responds to chemical signals, especially those given off by females. Chrysemys picta is found here in Lyon County and throughout most of the United States, and like most other vertebrates, it reproduces sexually. This means it requires both a male and a female in order to create offspring. Because of this and the difficulties that are associated with it, communication is necessary in order to find a mate for these animals. So communication helps produce the correct species in a mate and helps them find the correct sex as well. Communication in these turtles is mostly studied with tactile or touch communication or visual communication. Less often, people study chemical communication as well, which is the scent communication, smelling. This is kind of interesting because turtles are known to produce scent. They produce scent through urine and feces and different pores throughout their body. It's important to study communication, and especially chemical communication in turtles, because these animals are not doing so well worldwide as a clade. They're declining through overexploitation and habitat loss and road mortality. So understanding how this animal, who already reproduces slowly, reproduces and communicates is very important. Because of this, last summer I spent a lot of time in different ponds throughout Lyon County setting these different turtle traps. After I got enough of these painted turtles, I bring them back to the lab at ESU, and I set up a pool type enclosure here. These enclosures were divided into 3 equal sections, and a male painted turtle was put into the middle and allowed to swim around for 20 minutes. In each of these 3 trash bins, I had a male in one, a female in the other, and one remained empty. The trash bins had holes drilled into them so that chemicals could travel between the water inside and outside of the tubs, but the turtles can't see each other. And then for the 20 minutes, I recorded the amount of time the male turtle spent on each section of the pool. I did this for 27 different trials, and for each time I used a different male turtle in the middle and washed out the tub completely. And you can see my results on the table on the right. The most amount of time was spent in the female section on average, with 7.7 minutes, followed by the male section, and then the last section there is the control. I did a statistical test called a ANOVA test, and followed by a 2-keys test. And this illustrated a significant difference if you look on the left side of that column, a statistical difference between the female section and the control section. So the males really reported a better response to the female section than the empty section of the pool. All right, no statistical difference was found between the other 2 sections. However, you do see this trend if you look at the means. Okay, I hope you learned something about turtle communication, and thank you.
Sericea lespedeza is an invasive plant that's expanding its range throughout the grasslands of the Great Plains, displacing native grasses and reducing biodiversity. Grasslands once accounted for over 40% of global plant cover, yet 90% of the original North American tallgrass prairie has been destroyed. Invasive species such as Sericea cost the United States economy billions of dollars each year. Given the historical loss of grasslands and the economic cost of invasive species, it's critical to find methods of control. Annual spring burns are common in Kansas and other grasslands to reduce the abundance of unwanted trees and shrubs while promoting the growth of native grasses. However, this has actually been found to increase the germination and spread of Sericea lespedeza. We evaluated changing the burn season from spring to fall in conjunction with other common management techniques on the reduction of Sericea. So our research site was a prairie restoration with widespread and dense Sericea infestation. It was divided into 50 by 50 meter burn plots, including spring burns, fall burns, and unburned control plots, which were then subdivided into 4 sections, receiving a secondary treatment of herbicide, mowing, litter addition, or burn only. And in each subplot, 5 plants were evaluated each year by measuring biomass, stem count, stem height, and seed production. Our results indicated that fall burns in conjunction with mowing treatments had the biggest effect on reducing stem height, biomass, and seed production. From 2014 to 2015, the investment in seed production went down in plants experiencing fall fire or no fire compared to the investment in seed production for plants receiving spring fire. This is important because Sericea is a prolific seed producer, so anything that reduces seed output is going to be important for its control. Only herbicide effectively reduced stem count and resulted in 100% mortality of each plant monitored in the herbicide treatments. However, herbicide is expensive and harmful to the native grasses and forbs, as well as to the farmers and ranchers using this commonly for a control method. So we hope to provide land managers with tools to control Sericea while preserving the integrity of the tallgrass prairie. This would reduce the economic impact and environmental impact of herbicide use while increasing biodiversity and rangeland health. So this study was able to show that fall burns in conjunction with a mowing treatment can effectively reduce seed output and spread of Sericea without the use of harmful herbicides.